Our next Kavli lecturer today is Professor Xiaowei Zhuang from Harvard University. Xiaowei is the David B. Arnold Jr. Professor of Science, Professor of Chemistry and Chemical Biology, and Professor of Physics at Harvard. She's also an investigator at the Howard Hughes Medical Institute. Xiaowei is best known for her work in the development of stochastic optical reconstruction microscopy, or STORM, a super-resolution fluorescence microscopy method, and for her discoveries of novel cellular structures using this technique. She has been very well recognized for her outstanding research. Like Anna Maria, Xiaowei is a MacArthur Fellow. She won a MacArthur Fellowship in 2003, and since then, she's received numerous awards. I'll just mention a few: the ACS Award in Pure Chemistry, the Koblenz Award, the Max Delbruck Prize, the Sackler Prize. Xiaowei is a fellow of the AAAS and, of course, of the American Physical Society. She's an elected member of the National Academy of Sciences and the American Academy of Arts and Sciences. Just last year, she won the National Academy of Science Award in Molecular Biology, and she's a foreign academician of the Chinese Academy of Sciences. Please join me in welcoming Xiaowei Zhuang.
Not on? Okay, great. All right. Yeah, so I too would like to thank the organizers, in particular Sharon, for putting together a really wonderful session and for including me in this session. And after hearing the first talk by Anna, I realized how challenging this session will be for the audience, but since it covers an enormous scope. So I will, however, hope that you find the session fun too. So I will talk about the imaging method that we developed that allow us to look at molecular structures inside living systems. So with that, let me just start. I like to walk around, but I realize with 3 screens, perhaps this cursor works better than the laser pointer, so I'll just glue myself here. So as you can see, that cells are made of molecules. And these molecules, there's so many different kinds of biomolecules, and then they interact in very intricate networks, and together they give the cell its life. And with that, what my lab aspired to do is to find ways to directly visualize these molecular interactions in molecular processes in living organisms, and then hope that we can follow this model by Yogi Berra that you can see a lot by just watching. And through such a direct visualization approach, we get the direct knowledge of how these molecules act together to give us life. And when they go wrong, how do they cause diseases and so on. So if you look at these molecules inside the cells, there are a few properties. First is they're very small. They're nanometer scale. And second is they're jam-packed in very, very high density inside the cells, even almost reaching crystalline density in some cases. And then also there's so many different species, different kinds of atoms. So if you were to imagine an imaging approach that can tell you a lot about these molecular interactions, you would like your imaging approach to have these 3 properties. And that is, first, they need to have molecular scale resolution. And because these are nanometer scale subjects, you need to have nanometer scale resolution. And second, because there's so many different kinds of molecules, if you just have a black-white picture, of all the molecules together, not knowing their identity, then you're not learning a whole lot. So you would also like to have the molecular contrast or molecular specificity. And finally, because the cell is a living object, or any living systems, so there are many, many dynamics and changes that's happening together. And it is actually these dynamics that give us life. So you would like your imaging system or method to also have the dynamic imaging capability. So with these 3 things in mind, and then if you look back at existing imaging modalities, and you found that one imaging modality actually does pretty well, and that's light microscopy, in particular fluorescence microscopy. And that's because there are so many different colored fluorescent probes. Some of them are small molecules like organic dyes. Some of them are actually proteins that glows, called fluorescent proteins. And they can be linked to your molecular target with exquisite specificity. So with that, you can color with, you can combine with multicolor imaging and then use multiple colors to identify multiple species simultaneously and get your molecular specificity in your images. And of course, light is relatively gentle. In particular, today, we all want to go outside instead of sitting here because the sunshine is just absolutely pleasant after a long winter. I don't know whether the winter is as bad here as in Boston. So light doesn't easily damage the living systems. That's what I'm trying to say, although we do have to worry about light cost-damaging actual imaging. And because light microscopy also has very fast time resolution, when you combine those, you can get your dynamic imaging capability pretty well. Now, traditionally, what light microscopy doesn't do very well is on this nanometer scale front. And that is because, simply speaking, if you focus your light into a spot, and this spot is about 200 or few hundred nanometers instead of few nanometers. So that's 2 orders of magnitude bigger than you would want to have for resolving molecular scale interactions. And nowadays, we know very well that this is caused by diffraction. Light, as any wave, diffracts. So when you try to use a lens to focus your light spot, no matter how good a lens or objective you can find, the focal spot nonetheless has a finite size of about 200 to 300 nanometer in the transverse direction, or approaching a micron along the direction or the axial direction light propagates. So you use that, if you use that to scan your sample, even the smallest feature, like a single molecule, the image will expand into a blurry spot, and that is roughly that size. So that's why it's also called the point spread function. Now, if you have 2 subjects that are well separated, despite such broadening, you can still resolve them. However, as they get closer and closer together, eventually you reach the point their images so substantially overlap, what you see is a single blob. That's when you're no longer able to resolve that. And that's when you reach the diffraction limit. And that was first recognized by Ernst Abbe in the 1870s, and hence this resolution limit is also called the Abbe resolution limit. Now, nowadays, we know there are different approaches that can actually break this diffraction limit. And optically speaking, there are generally 2 categories of approaches. And actually nowadays, after we broke the diffraction limit optically, there are even ways to break it non-optically. For example, a clever way is to expand the sample, make the sample bigger, then you can actually use conventional optics to resolve even small subjects or objects. And that's, for example, called expansion microscopy. But I will focus on the optical ways of breaking the diffraction limit. And as I said, there are a couple of general categories. And the first one is using pattern-illumination to effectively shrink this point spread function of the microscope. And famous examples are stimulated emission depletion microscopy, or the STED microscopy, or its generalized form resolved, pioneered by Stephen Hale. And also a popularly used approach is the structure illumination microscopy, in particular in its saturated nonlinear form, also break the diffraction limit, pioneered by the late Mats Gustafsson and his co-workers John Sadat, Dave Agard at UCSF, and also by Rainer Heinzmann and co-workers in Europe. Now, the approach that I will focus on is taking advantage of single molecule imaging. So in the single molecule imaging field, we've known for a long while now that even though the image of a single molecule is still diffraction limited, means the width of the image is still 200 nanometer wide, nonetheless, you can pinpoint the center position of this image very precisely, roughly the width of the image divided by the square root of the number of photons you detect. So if you detect 10,000 photons, take that square root as 100. If you divide 200 nanometer by 100, that's 2 nanometer. So you can use that actually very, very precisely pinpoint the molecular position. That actually has been used as a powerful tool already to do biological investigation. However, that alone doesn't break the diffraction limit because, as I said, the diffraction limit comes in when these 2 molecules are close. So when their images overlap, how do you deal with that? And a biological specimen often is made of millions, not just 2 molecules, and their individual molecules, their images all overlap, give you a complex pattern, and how do you find the position of individual molecules? So that's where we came in. So about a decade ago now, we and others had independently come up with the idea of using the fourth dimension, what I usually say, or the time dimension. In other words, we try to separate these overlapping images actually in time, so that at any time or any snapshot, they're no longer overlapping. Now, that way, you can then pinpoint their center positions, and you can turn this subset off, and then you turn on a different subset, and then you pinpoint their positions, and you iterate this process often thousands to tens of thousands of times to determine all the molecular positions in your field of view. And then your image resolution is no longer limited by diffraction, but actually by how precise you can localize this molecule, which can be often 10 or 100 times better than the diffraction limit. So we started this because we actually, prior to this, discovered that some fluorescent dyes are photo-switchable. You can use light to actually switch them from dark state to fluorescent state. And if the light is weak enough, you get only a stochastic subset of them into the fluorescent state. So we call this stochastic optical reconstruction microscopy, hence the name STORM. Now, there are also parallel independent development by Eric Betzek, Harold Hazen, Jennifer Limping-Schwartz, and implemented their approach with photoactivatable fluorescent proteins and calls this photoactivation localization better known as PALM. I'm not sure I got all the words right. So, and then also Samuel has calls a similar approach FPALM. So here is how STORM is applied to this structure. You activate a subset of molecule at a time. You pinpoint their center position. Eventually, you get this kind of images. Now, these are some molecular tracks called microtubules inside the cell. And in the same field of view, this is what the conventional image gives you. So even without quantification, just by eye, you can already appreciate that there's a drastic resolution improvement. I will tell you the numbers later, but I want to get immediately into the 3D world because very quickly after we reported the 2D STORM images, we realized that we really actually need to break the diffraction limit in all 3Ds because the biological structures are often three-dimensional, actually mostly three-dimensional. So for STORM, that actually conceptually is quite simple. It means we need to add one more coordinate to the molecules, the Z coordinate. It's slightly easier said than done because the images that you take with cameras, they're flat. So how do you find the Z position? So when we first implemented 3D super resolution imaging, we actually used a very simple approach, and that is using a lens like that called cylindrical lens that only bent light in one direction, but not the orthogonal direction. And as a result, these single molecule images are elliptical instead of round. And the ellipticity depends very sensitively on the Z position of the molecules, and hence we can convert the 2D image of the microtubules into this kind of 3D images where the now Z codes the color codes the Z information. Now with that, you can actually take any part of it, rotate around, and then to see the real 3D perspective. So in this early work, the resolution we accomplished is 20 nanometer in XY. This is what I'm saying. You know, when movie plays, I lost my cursor. So it's 20 nanometer in XY and 50 nanometer in Z. So at this point, you can pause a little bit and say, well, this is 10 times better than the original limit. Hence, if I use this to look inside the cell, I will have 10 times better clarity, and I can learn a lot. And indeed, that's many of people in my lab do, and I will show you some applications. But if you're critical, you can also see that well, I set up the problem as I need a few nanometer in many general problems, not a few tenths of nanometer. There's still one order of magnitude to go. And there are some lab members that are that critical. Those are usually physicists, and so they want to actually improve the resolution. So I'll give you a few quick things, and only one example I will elaborate a little bit. First is we inserted the specimen in between two objectives, and with that we received twice as many photons, and also we have an error cancellation mechanism, and that actually improved the resolution by a factor of 2. But after that, you can still see that the Z resolution is still worse than the XY, and I like to have isotropic resolution. And so then we actually used this very interesting waveform that is relatively recently developed called Airy Beam. And that was brought into the lab by this very clever postdoc Xu Jia. And then in his PhD, he studied this waveform. So he educated me and telling me that this is a non-diffracting wave, just like Bessel beam, means that it doesn't expand a whole lot when it actually propagates. But in addition to the Bessel beam property, it also bends as it propagates. So he said that with the bending, I can determine the z position because molecules at different z would have propagated by different distance in order to reach the camera, so they would have bent by different amount. And then he come up with an approach to decouple this bending from the actual xy position of the molecules. Now, with that, you cannot taking advantage, not using the shape of the image to determine z, but to actually use the position, which you can be much more precise. And then indeed, using some spatial-like modulators in the detection path, you can actually come up with this kind of a bending beam. And on top of the bending property, you can see its non-diffracting property. This is the typical Gaussian beam. And you can see that out-of-focus light or out-of-focus molecules, by a few microns, you can no longer detect its light because it's so out of focus, while this non-diffracting beam actually still allows you to detect it so well. So with that, he gets isotropic resolution and also very large depth without actually changing the focal spot. And here is an example image he would get. These are some biological structures called mitochondria. You can see that the storm image is not only sharper than the conventional image. You can see a few of these mitochondria that are so out of focus that wasn't captured at all. You just don't see them at all in the conventional image. And now with the same focal plane, you can actually capture them with very high resolution. Then you can actually not only improve the resolution by physical means, you can also improve the resolution by chemical means because the number of photons we detect per molecule is a key determinant of the resolution. Now, if you can invent brighter fluorescent probes that give you more photons per switching event, you get a better resolution. And so indeed we did that with another very clever postdoc, Josh Wang. He developed a new photoactivation chemistry that allow you to get ultra-bright photoactivatable dyes that compare to our original discovery of photo-switchable molecules that has a 2 order of magnitude improvement in photon output. And with that, we can get resolution down to a few nanometers. And then indeed, being a light microscopy approach, STORM can also be used for live cell imaging in 3D in multicolor. And then we can get this kind of a resolution of about 20 nanometers in a second or less than a second time resolution. So with that, of course, being a fluorescence microscopy approach, which you could envision to apply it to a really wide variety of biological systems. And then here I just list a subset of our applications that range from relatively simple organisms. These are all single-cell organisms. This is ribosomes inside bacteria. And this is an interesting channel structure, ion channel structure in the sperm tail in collaboration with David Clapham. This is in collaboration with Sunny Shi, all the way to complex tissue samples. You don't have to just do cells or monolayers of cells. You can get a complex brain tissue sample and then see, for example, both the circuit level information of how synapses are organized in the neural circuits down to how molecules are organized in the synapses. And I want to actually just pick one example to illustrate the fact that these days we can not only use the super resolution approaches to get you prettier, sharper pictures, we can actually use that to discover novel cellular structures people didn't even know existed before. And I will give an advertisement that what I'm going to not touch today is some really interesting new application to how genome, DNA is organized in 3D in the nucleus. I'll talk about that in tomorrow's genome organization session. But today I'll focus on this new structure that we discovered in neurons. Now this structure is related to one kind of cytoskeleton molecule that's well known called actin. They form these, just like microtubules, these long filaments inside the cell and often give the cell its mechanical strength and a lot of other important properties. Now, for neuron is one of the what we call most polarized cell type in our body. It sends out processes. There are 2 kinds. One is called axon, and the other is called dendrite. And then the signal propagate down axon from one neuron to the dendrites to be received by the dendrites of the targeting neuron, so that these processes are very different. And actin is known to be important for this kind of a differentiation, or we call polarization of neurons, among many other important functions. So with that, you could see that it'd be very important to actually know what the structure of actin looked like in the cell. And there's very little knowledge because of the deficiency of imaging tools. So when we looked at that, in particular, we paid attention to what actin looked like in dendrites and axons, because these are the two very distinct processes in neurons that make them polarized, and it's extraordinarily important for the neuronal function. So in dendrite, you see this beautiful structure of long actin filament form these crisscross networks. And as I said often, as beautiful as it didn't tell us anything new. It's exactly how you thought actin should look like in cells, long filament forming this network. What surprised us and everybody else is what actin looked like in axons. You can see that in the same cell, just in the axonal processes, the actin took a dramatically distinct structure. Instead of forming these long filament, we still have that actually in axons, but the most prominent structure are these stripes. And they are so evenly distributed, you can even see by eye. And I should say that axon is a subject that biologists have been trying to image with high resolution, a bunch of different imaging modalities for almost a century, at least a half a century with advanced imaging approaches. And this structure is completely missed. And then if you want to understand why it's missed by conventional light microscopy, it's very easy, because if you turn on the conventional optics, that's what you see. You basically see a uniform distribution, so you won't uncover this structure. Now, this is a three-dimensional structure, so each one of the stripe you can turn around. You can see that these are actually these little rings, and these are the little rings that are right underneath the axonal membrane. And as I said, they look very evenly distributed. You could even computationally straighten out your axon and project the localizations and take a Fourier transform, and you get this kind of a remarkable peaks and harmonic overtones. And these together give you a periodicity of about 180 nanometer, which is below the diffraction limit, and hence it was not visible before. And it was interesting when I was talking to a reporter about this, they said, well, that's a literal skeleton. I've not thought of it this way. I think he was imagining the, dinosaur or fish skeleton and so on. So if you want to remember it, there is a skeletal structure inside the cell that looked like a literal skeleton. So the fact that they are so evenly spaced also give us some clue of what is making up this structure, because we envision there must be other molecules in this. And this other molecule has to satisfy the requirement of it has to link actin, and it has to be about 180 nanometer long to serve as a spacer so that you can have this even space. Now, that actually made us think of this molecule called spectrin. And the best known about spectrin in its ultrastructure is inside red blood cell, right underneath the red blood cell membrane. It formed this kind of a triagonal structure, and it is about 200 nanometer long, so satisfy the length requirement. And it does connect actin. So with that, we actually made this hypothesis that this is what this new structure looked like, a ring of actin filaments. And the reason that they're periodically spaced is because they're spaced by these 180 nanometer long spectrin tetramer. And if you want to test that hypothesis, you say, well, maybe I could also image spectrin. Now, if you get greedy, you say, well, I can not only image spectrum, maybe I could image a specific location of the spectrum, say the middle portion of the spectrum tetramer, and hence I would expect a alternating green magenta rings. And that's exactly what these talented postdocs, they were so not afraid of any of my requirement, and then they come up with these alternating patterns of actin and spectrum. So I will skip this part, and then just to show you that this is a very prevalent structure. This is an entire field of view. Other than this middle process is a dendrite. Every single axon in the field of view is having this letter periodic structure. And it's not only you can image it in fixed tissue, you can also see it in live cells. And these are in live neurons. You can see this periodic structure. And in closing this part, I just want to share with you a little bit of what a dramatic structure like that could do to neurons. Now, the first thing we noticed is it's right underneath the membrane. It actually anchors some very important membrane proteins in axon. And one group of very important membrane proteins are so-called ion channels, because it is the ions that flow through the ion channels that propagate in axon that actually give us the electric signal that is propagating in our brain, which is called action potential. So we found that these ion channels are actually anchored by this structure in a periodic fashion. So you could imagine that it actually influences how this very important action potential is established and is propagating. That's a hypothesis. A better known thing that actually has experimental evidence is on the mechanical front. Now, if I tell you that this actin ring is pretty rigid, but spectrum tetramer is very flexible, then you couldn't help thinking about a structure like that, right? Well, I don't know whether you couldn't help. We couldn't help thinking about a structure like that. Meaning that what you have is, I actually love this. I always say that, this is a thing that my daughter got it as a gift when she was born in 2010, and she loved it. And then since 2013, she never saw it anymore, because 2013 was when I start talking about the structure, and I always use this as my demo. Because I don't know who gave it to her. I don't know how to buy this kind of thing. So you can see that if you have flexible things, linking rigid rings, what you can come up with is a very elastic support of this tubular structure. For something as long and skinny as axon is, this kind of a flexible support is exactly what it needs as a mechanical support. And I'm not just saying that, because indeed, if you actually delete spectrum molecules from C. elegans, as has been done by Jurgensen and Bastini lab, what you found is axon tend to spontaneously break. And miraculously, if you can paralyze the animal, let it develop without moving, the axon actually grow in fine shapes and so on. So it tells you that this is a structure that fights against mechanical stress that the animal actually encounters. And so with that, let me actually just quickly switch gears to tell you some of our recent development of imaging approaches. Because as I set it up at the beginning, it's not just one or two kind of molecule that give us life. It is the collective behavior or collective interaction of thousands of different kinds of molecules that together give us life. So it would be nice to actually study all these molecules simultaneously. And we like to develop imaging approaches. So we think about how do we image all these molecules simultaneously. And we're obviously thinking along the line of DNA, if you hear about the talk tomorrow. But between RNA and protein, we pick RNA. And that's not because proteins aren't important. They're absolutely important. It's just because, as I confess, RNA is an easier thing you could imagine how to label them all simultaneously. And that's because it has these nucleotide sequences, and then you can come up with orthogonal or complementary sequences that can bind to them very specifically, and then at the end of this little oligo, you can attach a fluorescent dye, and this way you can very systematically label all RNA molecules easily. And that's called fluorescence in situ hybridization, FISH. That's easy to remember. And that's actually also a biological reason why we want to image all. I have a cold. I usually don't need the water in between. And why do we want to study all RNA simultaneously? That's because if you look at our body or any living organism, actually in our body you can see so many different kinds of cells, but their genome are actually more or less identical. The reason that there's so many different types of cells and so different behavior between different types of cells is because different genes are actually expressed to different levels in different cell types. So if you could imagine an approach to profile the gene expression at the systems level, then you have a way to precisely and quantitatively study the molecular basis of the different cellular behavior. And indeed, that's already known to many biologists, and then there's already very powerful approaches doing that, and that's called RNA sequencing or single-cell RNA-seq, which, actually the next speaker, Weiss is the expert in. I don't know whether he'll talk about it. So with such powerful single-cell RNA-seq, he said, why do you want to develop a different approach to do that, an imaging-based approach? And the reason is because we can get spatially resolved information. Because within cells, these transcripts or RNAs are not uniformly distributed. Their non-uniform distribution is actually very important for the local structure formation and maintenance. Okay, and also inside tissue, there's so many different cell types, and if you have to, dissociate all these molecules and then cell typing them, it is harder to get to know the knowledge of which types of cells interact with which. And if you could directly image that, you can get that kind of interaction network information. So how do we do that? As I mentioned already, we take advantage of this FISH approach. We use single molecule. Do I have a problem with my microphone? No, okay. So we use single molecule FISH, which is pioneered by Rob Singer, that actually gives you very high detection accuracy of individual RNA molecules, and perfected by people like Arjun Raj and Alexander von Woodner and so on. And then you can actually use multicolor imaging and multiple different color FISH probes to look at multiple types of RNA species. You could even imagine smart combinatorial approach to do colors to get 20 or 30 species of RNA simultaneously, as Singer, Raj, and Long Kai has been doing. But a transcriptome has thousands of RNAs. So we don't have this many colors that allow us to combinatorially get us 1,000 or several thousands. So what do? So we come up with this idea of using combinatorial labeling and sequential imaging. So I'll briefly talk about what it is. So for each different kinds of RNA, we encode it with a binary code 1010 and so on. In the first round of imaging, we actually only address those RNAs, their first digit should read 1 but not 0. So that means we label and image only those RNAs that first digit reads 1. And then we turn them off. In the second round, we label and image only those RNAs their second digit reads 1 but not 0. And you iterate this, and eventually this is the real image. And afterwards, you get this kind of image where the RNAs inside the cell look like dots, and they each have a string of binary codes attached to it. And the string is 1 for a digit if that round it's detected, and 0 for that digit if that round is not detected. And eventually it comes to this mathematical problem. After n rounds of imaging, how many different kinds of RNAs can you distinguish? You said, well, for n-digit binary code, how many different codes there are? And that's a very pleasant scaling. That's 2 to the nth. So that means a mere 16 rounds of imaging would give you 65,000 different RNA species. This exponential scale is really good. And 65,000 is the entire transcriptome of human, including both coding and non-coding RNA, except that is just too good to be true. And the reason is because of error. I said that single molecule FISH is very precise. You could even try to get to 5% error per. Okay, I'll take 2 minutes and then, okay. You can even take just 5% error per round of imaging. But after 2 rounds of imaging, that becomes 10%. After 3 rounds, it becomes 15%. And after 16 rounds, I can give you a transcriptome. But if you can be excited. And if I tell you that, well, 75% of the RNAs are misidentified, you say, whoa, bogus, what are you doing, right? So that's the problem. And how do you solve that? Luckily, that is actually a solved problem, just in an entirely different field, and that's called digital communications. Because our text messages, emails, and so on, these are all transmitted in binary codes, and they are very rarely having errors, and that's because they're not transmitted in normal binary codes, but error-robust code. And in essence, that means you take a subset of codes, and it takes multiple bits errors to convert one valid code to the other. So that makes the error chance, that makes the probability of making error or misidentification much lower. For example, in this famous Hamming code, if Hamming distance is 4, it takes 4 errors to actually convert one RNA into another. And that actually reduces the error to a very low value. And also because if you have only 1 error, you can correct that to the original RNA because it's at least 3 errors away from all the other RNA codes. So then you can also get high detection efficiency. We modify this to fit the FISH property, which I won't tell you how because I'm running out of time. In this modified Hamming distance 4 code, we can get 80% detection efficiency and less than 10% misidentification error. So we call this multiplexed error-robust FISH, or MERFISH. And I'll just tell you very quickly how well it works. With 16-bit, now modified Hammond distance 4 code, you can only code 140 RNA species, but we can indeed detect that with 80% detection efficiency and less than 5% error. And then we have also demonstrated detecting 1,000 genes by either increasing the bit number by keep the accuracy of the code, or by reducing the accuracy of the code a little bit, and then also use a fewer bits. And then in both cases, we get 1,000 genes. And then with that, indeed, we learned very interesting things about the spatial distribution of RNA inside the cell and gene regulatory networks. And with that, we actually imagine this approach is scalable to the entire transcriptome size of tens of thousands of genes. And with that, we hope to indeed reveal the distribution of RNAs inside the cells, and more importantly, the distinct cell types in tissue, both in normal tissue like our brain tissue, how many different types of neurons there are, how do they interact, and also in disease tissue, such as in tumors, such a heterogeneous environment, which cell types are drug resistant, for example, and which are not, by using this kind of imaging approaches. So with that, let me just close by thanking the people, and I would take too much time if I thank all the people who contributed to STORM development. So I'll just thank the people who are involved in the periodic structure. That's Ke Xu, Guixin Zhang, Zhang He, and yes. And then the MERFISH development, Jeff Moffett, Alistair Boutique, Kakao Chen, and then over the years we enjoyed many, many collaborations, which I didn't have time to really feature any one of them in detail, but I will do one tomorrow. And then thank Hughes and NIH for support, and thank you for your attention.
We have time for a question or two.
Question here.
We'll start with you right there.
You mentioned an imaging time of 1 second over here.
Okay.
You're right. You mentioned an imaging time of 1 second in some point, and I think of the cell as just in constant motion and things moving. At what time scale do things change position enough to blur your image?
Yeah, so that's a very good question. First of all, 1 second is just for that kind of image. For different type of structures, some would take longer, some would take shorter, okay? That's the first thing. And second is indeed some structures, 1 second is entirely possible to capture without substantial blurring. Those structures move very slowly inside the cell, but some move blazingly fast that 1 second it gets totally blurred. So again, it really depends on the systems that you're going after. Some needs millisecond time resolution, some actually even minutes time resolution would be sufficient.
Thank you.
Yes.
Oh, I just had a question about that. I was curious about that movie that you were playing at the beginning of your talk. Could you tell us something about
So
that?
that's an educational movie that's made by some Harvard, college teacher in together with Hollywood, movie rendering people and so on. So it's called Inner Life of a Cell. So if you do that search, you'll be able to get that movie.
Okay, so you're not quite able to see that in real time.
No. Yeah, that's a very good question. So I like that question a lot, because if we do have that kind of thing for every molecule inside the cell, I would just retire already. And obviously that's not accomplished. A lot of them were imaginations, although based on data, reasonable imaginations. Yes?
A really quick question. I didn't understand when you were doing the binary addressing of different mRNAs, how you did the addressing. How did you get only certain ones?
Yeah. So basically, if I explain FISH clearly, that means there are these oligos complementary to the RNA that we're interested in. So then we lay down all these FISH oligos to bind to all RNA simultaneously. But in this FISH oligo, we also have a part that sticks out. And then and we pick 16 different unique sequences for that. And for an RNA that its bit, binary code is called 1010, for example. And then it would have the sequence 1, sequence 3, and sequence 4 and 7, for example. And then I come up with a second round of FISH probe that only address those readout sequences one at a time. And this allowed me to actually label and image only those RNAs that bits reads 1 in that particular round.
Have you considered using your methodology for the eye, there's a tremendous need for in vivo measurements of the trabecular meshwork involved in glaucoma. There's almost no data on that. Have you considered working in that area? We
did some eye imaging, not that. We actually imaged neurons in retina, and actually that beautiful neuron image that I show is a type of retina ganglion cell. It is really an approach that can be applied to a wide variety of biological systems, so you're certainly welcome to use it. And then you can find collaborators, both, you can ask us, and then, we can show you maybe how to do the imaging, and they're better yet, you probably even have local collaborators, because STORM is a pretty widely, adapted method that many labs are using it now. And there's also commercial machine doing that, so you can buy a commercial machine and then do this kind of super resolution imaging.
Thank you.